Hello, and welcome back to Genetics Unsealed. I'm your host, Folabumi Adeleke, and today I'll be discussing an article and bioessentialism, something that sits at the crossroads of molecular biology, society, and philosophy. Bear in mind, I have to apologize, I do have a cough, so I will cough during this episode. Now, bioessentialism is the belief that our biology, and especially our DNA, defines who we are in a deep and unchangeable way. It's the idea that genes explain everything from our intelligence to our personality to the roles different groups play in society. It's neat, it's tidy, and it's completely wrong. In fact, a brand new study from Stanford and Stockholm University, published in Cell, provides a perfect lens through which to dismantle some of the biggest misconceptions surrounding genetic determinism. The research shows in exquisite detail how a transcription factor called KLF1, crucial for erythrocyte development, actually recognizes DNA, and as we'll see, the complexity of the mechanism completely undermines simplistic, essentialist narratives about genetics. So today, there's two goals: to unpack what bioessentialism is, why it's seductive, and why it's scientifically flawed. Second, oh, to explore how the new KLF1 research provides a real-world example of just how nuanced gene regulation truly is. Now, before we talk about transcription factors and DNA binding, we need to talk about the myths people attach to DNA. Bioessentialism assumes that genes are a kind of biological destiny, that they define who we are, what we can do, and how we fit into the world. You hear it all the time. There's a gene for aggression. There's a gene for intelligence. This group is naturally better at X, Y, and Z. It's an argument that's been used historically to justify sexism, racism, colonialism, class hierarchies, and eugenics. And it still shows up in modern debates about behavior, identity, and social roles. But the more we learn about genetics, the less the genome behaves like an instruction manual, and the more it behaves like a dynamic, context-dependent, deeply interactive system. Now, here are the core scientific problems of bioessentialism. Genes don't act alone, they act in networks. Most traits involve hundreds of thousands of genetic variants interacting with one another and with the environment. There's rarely a gene for anything. Polygenic. Gene expression is dynamic, not fixed. Cells constantly adapt to stress, hormones, nutrition, environment, and epigenetic cues. The same DNA can produce dramatically different outcomes depending on context. Genes set ranges, not destinies. A gene rarely dictates an outcome, it sets possibilities. Your environment determines where in that range you land. Social categories do not map to neat genetic differences. Humans share 99.9% of their DNA, and most variation exists within groups, not between them. Biological determinism about groups collapses on the even basic population genetics. Gene regulation is probabilistic, not deterministic. Proteins make decisions based on thermodynamics, chromatin structure, competition, cell state, and physical context. This all makes the new KLF1 study so relevant. It doesn't simply tell us something about erythrocytes. It reveals how our genome is actually interpreted and how misleading the concept of biological inevitability really is. So the new research from Stanford and Stockholm University, a team led by first author Julia Shape, a PhD student in the Greenleaf Lab at Stanford, along with researchers including Emil Markland, assistant professor at Stockholm University and Sci-Fi Lab, set out to examine how a key transcription factor, KLF1, actually recognizes DNA in human cells. KLF1 is essential for the development of red blood cells, the cells that carry oxygen around the body. It binds to DNA to turn specific genes off, on or off during blood cell formation. When this process goes wrong, it can lead to disease, including certain cancers. The team developed innovative, high throughput methods to measure how KLF1 interacts with DNA in vitro, in carefully controlled test tube experiments, and in vivo inside living human cells. The result? The behavior of KLF1 in living cells matches the behavior observed in vitro. As Markman put it, the most important result is that we show that it is possible to understand the binding between the transcription factor and DNA in human cells, and that this behavior is consistent with what we measure in test tubes. This is not a technical achievement. It's a foundational insight into how gene regulation works. One of the most fascinating findings comes from Julia Shape's work. Traditionally, we thought transcription factors recognize short specific sequences of DNA, a kind of molecular postcode, but the new research shows something far more sophisticated. Here's Julia. We discover that this transcription factor pays attention to much more of the DNA sequence surrounding its binding sites than previously thought. This means KLF1 doesn't just read the key letters of its binding motif, it reads the context around them. This context can strengthen or weaken its decision to bind, and these decisions help encode gene regulation in the genome. It's not just sequence equals function, it's sequence plus shape plus surroundings plus chromatin plus thermodynamics equals function. Gene regulation is an interpretive act, not a rigid mandate. Markland mentions another critical point. When transcription factor binding goes wrong, diseases can emerge. This is especially relevant because more than half of all mutations linked to genetic diseases occur in regions where transcription factors bind. That means disease often comes not from broken genes, but from miscommunication between DNA and the proteins reading it. Think of DNA as a series of road signs. The transcription factors are the drivers. If the signs are unclear, or if the driver misreads them, the consequences can be serious. But this also means traits and diseases do not arise from single genetic factors acting in isolation. They emerge from interactions, from probabilities, from context. And again, this complexity undercuts the simplistic deterministic narratives at the heart of bioessentialism. So, what does a study about KLF1 and red blood cells have to do with society level narratives about genetics? Everything, and here's why. The genome is not a set of fixed instructions. Transcription factors read DNA conditionally, not mechanically. DNA's meaning depends on its context. Genes only matter because of how cells interpret them, and interpretation changes constantly. Biological systems are probabilistic. There's no single deterministic path from genotype to phenotype. Gene regulation is not destiny. Your environment, experiences, nutrition, stress levels, and even social circumstances all influence how genes are expressed. Genetics can explain variation without dictating identity. Genes shape us, but they do not define us. That's the heart of the matter. If bioessentialism says your DNA makes you who you are, Modern molecular biology answers your DNA participates in who you become, and that is a much deeper, more accurate, and far more humane understanding of biology. The study, titled Thermodynamic Principles Link In Vitro Transcription Factor Affinities to Single Molecule Chromatin States in Cells, gives us a more complete feature of how transcription factors operate in the complex real complex real complex environment of the human nucleus. But beyond the technical achievement, it gives us a reminder. Biology is powerful, but it's not absolute. Genes influence, but they do not dictate. DNA is important, but it is never the whole story. Understanding the limits of genetics, not just its capabilities, is essential for responsible science and for responsible conversations about identity, health, and human difference. That brings us to the end of this episode on Genetics Concealed. Today, discussed bioessentialism and a new article that has recently come out. That's it today. Your host, Folabumi Adeleke, signing out.